Pulmonary arterial hypertension is an irreversible disease with no known cure. One form of reversible pulmonary hypertension occurs when breathing low oxygen, or hypoxia. In response to hypoxia, there's a combination of increased blood flow and reduced pulmonary vessel diameter, resulting in an increased pulmonary pressure, which could lead to right heart dysfunction. Elite breath hold divers are those who routinely hold their breath and place themselves in severely hypoxic conditions. These individuals may therefore have altered responses to hypoxia, which may help to better understand pulmonary hypertension. Thus, the purpose of the study was to 1. Determine if pulmonary arterial pressure and right heart dysfunction in hypoxia was greater in elite breath hold divers compared to controls, and 2. Determine the role of a pulmonary vasodilator to minimize the increased pulmonary pressure in response to hypoxia. This study was a single-blind placebo-controlled trial involving 26 subjects, consisting of 13 elite breath hold divers, each compared to an age and sex-matched control. See Table 1 for anthropometric data. All subjects had normal heart and lung function. Study Day 1 consisted of subjects taking either a tablet containing 50 milligrams of sildenafil, which is a pulmonary vasodilator, or a placebo. Both the subject and the ultrasonographer were blinded to the treatment type. 1 hour after oral ingestion of the tablet, subjects were instrumented and began breathing on a system designed to control oxygen and carbon dioxide levels in the blood. The subject would breathe room air for 5 to 10 minutes to establish baseline values, followed by 30 minutes of hypoxia equivalent to 17,000 feet of altitude. Measurements of pulmonary pressure, heart rate, and cardiac output were made, and right heart function assessed via tricuspid annular plain systolic excursion. The total pulmonary resistance was calculated by dividing the pulmonary pressure by the cardiac output. Stroke volume and cardiac output were normalized for body surface area to account for differences in body size between subjects. Subjects returned after a minimum of 48 hours to repeat the study with the alternate treatment. See graphs 1A through 1E for the results of this study. We observed a statistically significant difference in stroke volume between the divers and controls at baseline with sildenafil and at 30 minutes of hypoxia with placebo. There was no statistically significant change in heart rate between the two groups. However, there was a trend indicating that divers had a lower heart rate. Because there were no significant changes in cardiac output, this indicates that divers potentially have a cardiac adaptation that allows them to deliver the same amount of oxygenated blood while expending less energy. Additionally, we observed a statistically significant difference in total pulmonary resistance between divers taking placebo versus divers taking sildenafil at baseline. This could indicate divers have chronic pulmonary vasoconstriction in room air from repetitive intermittent exposure to hypoxia. We observed no statistical significant differences in cardiac output, pulmonary arterial pressure, or right heart function at any time point with either treatment, indicating no significant cardiopulmonary dysfunction. In conclusion, despite repetitive exposure to low oxygen, the breath hold divers had heart, lung, and vascular responses to hypoxia that were comparable to controls, indicating no maladaptation. I'd like to thank Dr. Andrew Lovering and Tyler Kelly for assisting with this project, and the Burroughs Wellcome Fund, along with the Fulbright U.S. Scholars Grant for funding this project.